You're here.
Yeah, thank you. So I'm actually from Thomson Reuters, but I was a full-time employee consultant for Boehringer Ingelheim, involved in the involved in the define and design, testing, data curation for the Transmart at Boehringer Ingelheim. And other people I wanted to acknowledge are Eugene Rahmatulin, Alex Ishkin, and Sirimon from our side, whom many of you know. And from BI side, it's Yirong Wang and Will Logging. And so, yeah, there's a lot of smart abbreviations there. And we named this workflow-based Transmart plugin BSmart. So, yeah, no pun intended. So, as you probably know, there are basically two types of users who are using Transmart. One is hardcore bioinformaticians, computational biologists, and another just regular biologists and scientists, right? And so we had a pilot project at BI in 2012 where we curated 12 immunology data sets. That was for the BI inflammation and immunology group. And so they had a chance to explore it, and they said that it's a, Transmart is a beautiful platform that allows you to generate all this research hypothesis by studying the correlations between genetic and phenotypic data. Yeah, but in spite of all its beauty, it's a little bit hard to understand. So they raised some concerns and asked us if we can develop something to, help address those issues. So the main issue that they have is they wanted the ability to work with across several data sets at the same time, right? So as you know, Transmart works just with one data set at a time, right? And so we were able to overcome that, and that's, I consider that a major breakthrough in Transmart, but there are also other issues that were identified. So I was thinking it's not exactly user-friendly, so we basically created this a three-step workflow, that allows them, that precludes the wrong choices that they can make. Obviously, the steps were not very easy to remember. They would do it real fast after initial training, but let's say that was there actually phrasing that, we come back in 6 months, we try to do it again, and, we don't remember anything. So we incorporated some help hints and saved workflows for that purpose. Obviously, drag and drop is very powerful for cohort selection and provides a lot of freedom. But sometimes, when you have too much choice, it's actually hard to choose things and it becomes frustrating. So again, they wanted some sort of checkboxes and radio button interface, again, to preclude the wrong choices. And obviously, everybody likes cartoons, right? And Transmart has no interactive visualization at all. So we did some interactive visualization designs for them, and we simplified the Excel export. All right, so the data curation is the same, right? We start with the raw data to load the studies, do all this pre-processing and normalization. The ontologies are really crucial here when you're working with multiple studies at the same time, right? Because that allows them to be matched, right? So, for example, if there is some diabetic nephropathy study, right, and one parameter named in one study would be like percent of glomerulosclerosis, and in another glomerulosclerosis percent, and obviously you won't be able to match these two studies based on that parameter. So it all had to be standardized, to use all that unified terminology. And so, BI scientists, they basically proposed this 7 questions that they want to get answer to, and basically better interface, and you'll see how it looks a little bit later. So some technical details, it was implemented using the same Groovy Grails technology as our original Transmart. So it basically goes on top of Transmart. This collapsible accordion style for workflows. Again, you'll see how it looks later. For the interactivity, we use JavaScript D3 libraries. Obviously, we cannot build a software and not connected to Metacore pathways, so there is a link there. Then yes, the content-based help and preview to make it easier for users to understand the analysis process. Again, workflows can be saved for future analysis, and results exported easily into Excel and PDF. So these are the same workflows that they came up with, but they basically come down to two use cases. One is you start with a known genes of interest, basically with some sort of gene list or gene signature. You load that and you want to explore expression patterns of those genes depending on different clinical parameters. And the second use case, you basically you don't know anything. You just start with a data set or several data sets, and you want the workflow to generate the best gene signature that would discriminate between two cohorts. For example, disease and control, responder, non-responder, maybe some doses, etc. So that pretty much summarizes this 7 workflows. We had to use a lot of flow charts to actually understand what kind of output they want to see. For example, if you start with gene signature and one data set and no parameters, how visualization would look like. If you then select a categorical parameter or numerical parameter, how it would look like for one data set. Then if you choose several data sets, how would that change the visualization with parameter or without parameter? And then obviously, if your gene signature is just a couple of genes, obviously you cannot build too good of a heat map for 2 genes. So then it's some sort of box plots there. So this is how the interface looks like. So it's basically like a extra tab in the Transmart, right? This is this accordion style workflow selection. So when the first step is open, it allows you to select those 7 workflows. If you click next, then this would fold and the next step would open. This is the customized saved workflow that you can select. If you click on this, you will see other workflows that can also be deleted from there. This is this help hints window, right, to help remember what to do, and the preview showing what choices were made during each step, and then you basically save that as a custom workflow. So once you selected the workflow, you click next, go to the second step. This is where you either select your genes of interest, so it can be either the gene signature from the Transmart already saved, or you can just type any gene and add it there, and it can just pile up like that, and then you can remove it if you want. And if you, or you can just work with a data set, right? So that's just a checkbox switch. So then you click next to go to the third step. This is where you select the database of interest. So there's like a filter to select if you want to work with animal data sets, clinical or cellular. There's this lookup window to type for some text to enable you find the data set faster. So when you click on it, it gets selected, and the selection is showed here. You can remove data set if it's the wrong one. There's this information icon. You click on it, and it opens the metadata about the data set. And then, again, once you select the data set, it shows clinical parameters annotated for that data set. And so if it's 2 or more data sets, it only shows common parameters that are again, common between those several data sets, right? So again, that's why the ontology and all this vocabulary is very important here. So once you've done all your selections, you click next and it starts the analysis. So here is how the result page looks like. This is the heat map, right? This top panel labels the different groups within the data set. So yeah. Then if you click on one cell, it will show this kind of statistics for a gene per sample, right? And show minimum, maximum, and median value. And then you can select it as a square like that, and then this table will be updated. And then you can actually move that square around, and it will change this table. So that's the interactive part of it. And whatever selection you make, again, it will be shown on a table, and then you can save it as a signature in Transmart. You can export the results either as Excel file with all the detailed gene information, or this picture as a PDF file. And again, the workflow can be saved, and you can, when you click finish, you go back to the first step where you can select that custom workflow again in 6 months. And yeah, for whatever gene you have selected, you click this run enrichment button, and it launches this enrichment analysis, basically using the Metacore Thomson Reuters pathways, right, showing pathways for your selected gene signature. And yeah, if you click on any pathway of choice, it shows our signaling pathway maps, right? And again, for those who don't know, each icon shapes represent different protein types, and then everything is color-coded, or interactions, the effect is color-coded, and then you see these mechanisms in these hexagons in there, and it's all interactive. All right, so that was just the interface, how it looks, and this is the actual use case. So we chose one psoriasis study. They did a study on patients. They took lesional and non-lesional skin, right? And so the first question we had would be, if the workflow can, generate a signature that best distinguishes between lesional and non-lesional skin, right? So this is obviously the result. You see that there's a number of genes that are upregulated in lesional and downregulated in non-lesional, and the other way around. So we also wanted, if you do this analysis with slightly different protocols, it always gives you slightly different results from the paper. So we wanted to compare it to the paper, and basically, if you look at the top 50 genes discovered by this BSmart, it's basically 56 of it were in the top 50 in the paper, and then top 100 genes discovered by BSmart, they contain 78% genes that were mentioned in the paper. So that was a pretty good agreement with the published results. Now, the next question we had is actually to see how the signature performs in other studies. So we picked 2 more studies that have patients with lesional and non-lesional skin. So the second psoriasis study comes from the same lab, and you can see that, for this initial study from which the signature was derived, this Suarez study, right, there's this real nice pattern separation, and the pattern stays pretty much the same in the different psoriasis study, well, except maybe for these genes, which expression is not very clear. But then if you look at the scleroderma patients, again, lesional versus non-lesional skin, there's no pattern at all, right? It's totally random. And then if you look just at the scleroderma patients alone, you can see that there's basically no pattern. The signature has no pattern, which, again, if you compare to the expression pattern, how it looks here, right, which is clear and basically random noise in scleroderma. So this just suggests that scleroderma perhaps has other pathology mechanisms, right, or maybe something's wrong with the data set. So we can, just by looking at the data, come to some conclusions about even the data set quality, actually. So this was analysis of the, skin expression. And the last study we did, we wanted to see how this signature derived from psoriasis, lesional, non-lesional skin, if it has any expression pattern in other tissue types. So we got this ulcerative colitis study with intestinal epithelium, right? And so again, it's the same signature from that psoriasis study. And again, it was a very nice study. It has the active ulcerative colitis samples control, then non-active samples, basically, which do not have any lesions, and then the ulcerative colitis with remission. And so again, some genes that were overexpressed on lesional skin were also overexpressed in the ulcerative colitis active patients. And again, the ones that were overexpressed in control were also, in psoriasis skin were overexpressed in the ulcerative colitis control. Non-active ulcerative colitis samples were pretty much like control, which is good. And then you can see that these genes in remission samples, they actually somewhere in between the active colitis and the control, right? So basically, it's in good agreement that, although there is no more disease, but it's not exactly normal either, right? So this analysis was done pretty, fairly quickly, within an hour, right? We just went through all these several data sets, played around, and just generated that very nice signature from this. To summarize, in BE-SMART at Boehringer Ingelheim, there are about over 100 curated data sets. A lot of it public, some private data sets, both from gene expression, and there's NGS data loaded in there. Again, this is a very nice framework that can be used to create other workflows. The system is designed for biologists, right, lab scientists, and just for them to play around, discover any patterns, and then if they have any questions about what they discovered, they go then to the statisticians and computational biologists for in-depth confirmation of their findings. Obviously, Transmart 1.2 has been released, and we already have upgraded BE-SMART to the 1.2 version. So some modifications were done there. And yeah. So this is a computational biology group from Boehringer Ingelheim. This is Yirong Wang and Thomson Reuters. We don't have a picture, but you can meet us live here. So thank you.
Hi. Do you have any plans to do this type of workflow for, say, proteomics or metabolomics in the future?
Proteomics, metabolomics. Well, the workflow doesn't really depend on data type. It's if you're able to load it into Transmart, then it can run on that.
Okay, and just one other question. Does your heat map rescale if you have a lot of genes?
Yes, they all change, and sometimes when you do several data sets at the same time, depending on the settings, right? Well, actually, if you haven't selected any parameters, then it would just show the heat map, so sort of merged for all the data sets, and you can see them all at once. It's just there was a request to also see individual expression and then the fold changes, so it was hard to reconcile. So, yeah.
Hi. Can you elaborate a little bit more on how you enable the cross-study analysis?
Cross-study? Yeah, it basically uses the z-scores, right? That's, Transmart calculates the z-scores, and that basically normalizes the data set. And so that's what allows you cross-study comparison, because they're all, centered, right? And the deviation is standardized. So that's what enables the comparison. Okay?
So it means you have renormalized across all the data sets?
Well, yes, but that's how it is. When you load data in Transmart, right, it's, the process data, they are looked to normalized. signals, right? But then Transmart converts it into z-scores, and even Transmart results, you see them in z-scores. Yeah, it's all normalized, yeah.
Thank you. Thank you for a nice presentation. A couple of detailed questions about your implementation. When it comes to the workflows that are implemented, are those can you expand those and add new workflows without changing the underlying code base, or is it configurable, or is it codable and extensible?
Well, yeah, and that's what it says, that can be more different workflows can be created on the same framework. Yeah, so
But you would write code to do that, or you would be able to?
Yeah, you have to write
not a data-driven
extra code. Yeah,
configuration.
some maybe slight modifications, because it's also you have to maybe change our scripts and then change the visualization. So yeah,
so it's really,
you have to do some code additions.
And I guess the other question is this something you mentioned you'll be porting it to 1.2? Is all of the capability that you've created available as open source?
There are plans to release it to the community, but it might take some time.
Might take some time. Thank you.
So and it's for BI to decide.